Hello, everyone. Today, I want to talk to you about nanotechnology. Now, the basic idea behind nanotechnology is that, okay. The basic idea behind nanotechnology is that we can use the special properties that materials have at the nanoscale to accomplish things that we wouldn't otherwise be able to accomplish. So nanotechnology, in the not-so-distant future, will enable us to create batteries which can last for weeks but charge in just seconds. It will also allow us to create cars, buildings, and even clothes which never need to be washed because they just don't get dirty. Now, when we start using the basic principles of nanotechnology to solve problems in human health and disease, then we enter a field known as nanomedicine. Now, when you think about it, all of the basic functionality of our bodies, all of the basic machinery of our cells is at the nanoscale. So when we talk about proteins, enzymes, nucleic acids, all of these things are on the nanoscale and function at the nanoscale. So to put it in other words, Mother Nature operates at the nanoscale. And if we want to beat Mother Nature, we too have to operate at the nanoscale. So what does this look like? What does this mean working at the nanoscale? Well, if you read a bunch of science media, you see pictures that look something like this. Now here we have nanoscale robots, which are designed to patrol the bloodstream and to attack disease the moment it arises. This is a little bit unrealistic, and frankly, it might be impossible. But that doesn't mean nanotechnology is a pipe dream or that it doesn't exist. It just looks a little different. So when we talk about nanomedicine in terms of today's technology, what we're really talking about is nanoparticles. Nanoparticles are just very small pieces of matter. They're just particles that are on the nanoscale. And when we talk about nanoparticles used in medicine, they look a little something like this. So here in blue, you can see the core of the particle, or the nanoparticle itself, and then it has this red and white coating on top of it. The purpose of this red and white coating is to modify the interactions between your body and the particle. So essentially, you can think of these particles as very tiny capsules. And when we think about them this way, we get into another concept, the concept of nanocarriers. So now we have a payload, for instance, a drug, and we have a surface coating which changes the way it interacts with your body. So it can protect your body from the drug and protect the drug from your body. And this gets us to probably the most important concept in nanomedicine, which is targeted nanoparticle drug delivery. The idea here is that conventional medicines are, when injected into the body, distribute themselves relatively uniformly throughout your body. Now that's a bit of a simplification, as you probably well know, but for the purpose of this talk, it's close enough. Targeted nanomedicines, on the other hand, can greatly increase the dosage at the local target site and reduce the dosage in the rest of your body. So these particles have the potential to simultaneously improve efficacy of your drugs and reduce potential side effects. So I'm sure you can imagine how that could be a very important breakthrough. You're probably wondering at this point, where are my nanoparticle drug delivery systems? Where are my magic cures? Well, as it turns out, for this, like many things in life, and just about everything in science, it's easier said than done. One of the issues that we found for this kind of technology is what I like to call transport obstacles. So when you think about the path a nanoparticle has to take from the site of injection to the site of action, there are a number of obstacles that have to be overcome for it to get there and get there intact. Here's an example of one of those obstacles. This is extracellular matrix protein, specifically collagen. Now, it turns out that collagen is a major impediment for nanoparticles used as drug delivery systems. The issue with that is that collagen is found throughout your body, so it can pose a major obstacle. Now, this is an electron microscope image of collagen, And this gives you an idea of what it looks like from the particles perspective on a very small scale. So putting it on more human terms, you can think of the particles moving through these gels as you trying to escape from a forest that looks like this. You can imagine that while you probably would be able to escape from this forest, it's not going to be easy, and it's certainly not going to be quick. And that's what I study in my research. I look at how nanoparticles move through extracellular matrix and extracellular matrix gels. And we found a few things in our research. The first thing we found was that the density of the collagen matters. So you can imagine that if this forest had a few less trees in it, it'd be a little bit easier to get through. Size of the particles is also important. So if you picture a small child of 9 or 10 trying to escape from this forest, they probably have an easier time than a big guy like me. Furthermore, what we found is that the coating on the particle's surface impacts its ability to move through these gels. So going back to my forest analogy, you can imagine that these two people would have a very different experience escaping from that forest. So we've been able to find out what influences the way particles move through these gels, and we're able to come up with a suite of particles which can be tuned in terms of their transport through these gels. Now, not only does this allow us to design particles which can move better through these obstacles and better overcome these obstacles, but knowing this information, we can actually turn it around and turn an obstacle into something that's to our advantage. Let me give you an example. This is a histological image of an atherosclerotic plaque. Now, as I'm sure many of you are aware, atherosclerotic plaques often consist of a lipid-rich core with a fibrous cap on top. Turns out that this fibrous cap is extremely important for predicting patient outcomes because you can get thrombosis and even death if those plaques were to rupture. So understanding when these plaques will rupture and what determines their rupture risk is extremely important. And since we know that fibrous caps, their strength of fibrous caps depends on the collagen content to a certain extent, and we know how these particles move through collagen gels, then by administering particles to a patient and looking at how they distribute themselves within these caps, we can predict collagen content and even rupture risk. So now, not only can we get an idea of the size and the geometry of the caps, but even their molecular makeup. Let's talk about another obstacle. These are endothelial cells. Endothelial cells line the interior of your entire bloodstream. They line all of your blood vessels. And it turns out they like to take up nanoparticles. Now, if endothelial cells, because they're so numerous, are taking up a lot of these nanoparticles, then there's not going to be a lot left to get to their target site. So this can be a major obstacle for nanoparticles. But again, by further studying the problem, we've come to understand that these particles tend to accumulate in endothelial cells that are exposed to lower shear stress as opposed to those that are exposed to higher shear stress. So in high flow blood vessels, we find that we get less accumulation of these particles than in lower flow blood vessels. And we can even go one step further and say that we find that there's higher accumulation of particles, here seen in blue, in the lower shear stress regions of a single vessel than in the higher shear stress regions. Now, how can we take this knowledge about an obstacle and again, turn it around to our advantage? Well, one important pathophysiology is that tumor vessels are known to have a very disorganized and immature structure because of rapid angiogenesis. And we know that the flow inside these disorganized vessels is typically lower and weaker than the flow inside normal vasculature. So by encapsulating anti-angiogenic drugs into nanoparticles, we can actually get selective delivery to tumor vessels and administer the anti-angiogenic drugs only where they're needed. Nanotechnology is going to revolutionize our lives. It's going to change the way we view technology and the way we see the future. And that includes nanomedicine. things like nanoparticles and nanocarriers will become ever more present and ever more important, along with a whole host of other nanoprefixes in development to bring real change, real progress, the nanorevolution. Thank you.
Great job. Great job, Chris. That's very interesting. So we're going to take it open for questions here. If there's any questions in the audience about nanotechnology for Chris. While we're waiting, I'll start with one. How far away are these from actually entering the world of human trials?
So nanotechnology in simple forms already exists. So the first nanoparticle drug delivery system for delivering doxorubicin to tumors was approved in 1994, I believe. So there are more of these that are approved every day and more of these in development every day.
Yeah. Can you outline, sorry, thank you for your speech. Can you outline what way the endothelial cells are taking these up? Is it endocytosis? Is it a targeting antibody?
So the particular particles which were used in that image are what are known as non-targeted or passively targeted particles. So they have no active ligand receptor targeting. So those would be taken up by likely primarily endocytosis, but there's also a few other mechanisms that are possible.
Hi, really interesting research. I know that, as you say, they're using it in cancer tumors. Are there other areas that they are now working on a little more?
Yeah, absolutely. So the early research in nanoparticle drug delivery focused on tumors, and there's some very good reasons for that, including the leakiness of tumor vasculature, which allows accumulation. But tumor vasculature is not the only angiogenic pathophysiology. So we can look at things like atherosclerotic plaques, which also have angiogenesis and are characterized by angiogenesis. So there is potential for delivery there, and there's lots of particles in the works for that. So there are also more generic particles, for instance, iron oxide contrast agents that are nanoparticles, which are used for a range of different applications. But primarily the ones on the market today, to the best of my knowledge, are for tumor applications, but the research is for really any disease you can think of that would benefit from selective targeting. There may well be. I saw a presentation a couple years ago now on nanoparticles which present antigens, and they modulate the immune system, and they actually can dramatically improve the symptoms for diabetes and things like that.
So one last question is all we have time for.
Thank you very much for introducing this fascinating technology. You said before that there is more like a passive agglomeration in endothelial cells. Cyclodextrins, for instance, or other nanoparticles for drug delivery would use also probably receptor-associated kind of like uptake. Do you have any kind of expertise where this is leading in the future? Is it the same way? Is it a different mechanism than the one depending on shear stress?
So if I understand your question correctly, you're asking me the future for actively targeted particles, for ligand receptor specifically targeted?
The difference, predominantly where it could lead to.
Mm-hmm, mm-hmm. So active targeted particles are a very exciting field, and there's a lot of research into how those particles interact with cells, how they're taken up by cells, and how they can improve delivery to cells. The issue with active targeting is that it requires you to get extremely close. You have to have your ligand bound to the surface of your particle, able to interact with your receptor on the surface of the cell. So we're talking about nanometers apart, right, or even less. So those kind of scales are pretty difficult to achieve. So while there's a lot of very promising research early on to try and get the particles to go into cells. They haven't moved past in vitro, really, because of the fact that you have to get them to that close proximity with the cell. So you have to get them through extracellular matrix, outside the blood vessels, all of these other transport barriers. But there's no doubt that active targeting particles will be a crucial part of this nanoparticle drug delivery systems, because they are taken up in so vastly greater numbers.
I think we're going to have to put Chris out front to field questions over the break, because obviously you've piqued everybody's curiosity with this. So thanks again, Chris, a wonderful presentation.